My name is Simon Tavare. I'm the director of the Irving Institute for Cancer Dynamics. We were founded with the aim of enticing scientists and humanities people from the main campus at Columbia to work on cancer research.
We want to combine quantitative and mathematical sciences such as statistics, computer science, machine learning, development of genomic technologies with biology and cancer biology in particular.
We rely on collaborations with other people. We manage to place machines or devices that measure the things we need to measure in other people's labs, and then we use them, and we're then responsible for looking at aspects of the data. How do we interpret this information that we've collected? Most of these data sets are now extremely large in volume and very complicated, and so that's where we come in, trying to develop both theoretical methods and applied ones to understand what these data tell us about cancer evolution.
Cancer cells can often exhibit abnormalities in many different ways, and we also know that the cancer cells can interact with each other and the environment pressure in different ways. So boiling all of that down into a simple model is not always straightforward. Recently, we introduced CINA, which is a general approach and simulation method that can help us analyze the effects of different mechanisms of chromosomal instability in cancer evolution. We built CINA to be an efficient method, and that can incorporate new findings from the oncology community about new mechanisms of instability.
A fundamental problem broadly speaking, in genomics is matching. I look at your cells when you're 20 and when you're 50. They're going to be similar because it's you, but they've changed. How have they changed? Well, that's a matching question. So what optimal transport is about is a computational method for solving for these matchings. You are turning one probability distribution into another. Optimal transport, geometric data analysis more broadly, honestly, the whole paradigm in the literature of doing dimensionality reduction and clustering, all of this tells you that this data, genomic data, has shape and geometry, and that understanding that is clinically and scientifically significant. But it's also clear that it is not any kind of geometry we've ever seen before. And I think that's super exciting from a mathematical perspective, a computational perspective, and I expect also from a clinical and scientific perspective.
A big focus of our work is actually not plug and play AI and machine learning or statistics for applying it to genomic data, but to actually rethink and redesign these concepts in order to optimize interpretability and not just predicting if a patient will respond to a treatment or not, but being able to tell the clinicians why we think they won't be responding. And also, if we have a better understanding of the mechanisms, we can also design novel therapies that target the pathways that lead to cancer relapse and resistance to treatments. The same for metastasis. If we have an understanding of why this tumor might have higher probability of metastasizing to the lung versus the brain, then we can tailor their therapeutic treatment and adapt this treatment as we monitor the evolution of the tumor as well.
The way that we're studying extrachromosomal circular DNA is using single-cell DNA sequencing. The tool that we're working with has the advantage of having reads that are sort of proportional to the number of DNA fragments that are there, so it allows us to very easily count the number of these extrachromosomal circular DNAs that are in every single cell. Because ecDNA are inherited randomly and you can have many copies, if a cell has an extrachromosomal circular DNA that contains that gene, it will now have up to 100, 200 copies of that gene. And so what that means is that the therapy that was going to be targeting that gene now is not effective on those cells that have high numbers of extra chromosomal circular DNAs. And not only that, it will kill off all the other cells that have low copies of extra chromosomal circular DNAs. So now your tumor has been repopulated with these drug-resistant cells.
Wound healing, which is one of the areas of interest in my group, is a very complex dynamic process that involves participation from many players. So cells divide, migrate, read in signals from their environment, and to understand how to model this information in order to improve wound healing outcomes in real patients, we need to come up with better ways to test this in silico. We need to understand what happens at very small scales, like what proteins do, how gene expressions are being read and expressed, and how that in turn affects more macroscopic behaviors. This multi-scalar property is something that is an opportunity from a statistical point of view, but it's very challenging to observe at the experimental level because we can't look at all these properties at the same time. So we can measure maybe gene expression, or we can measure live imaging of how the cells look, but it's very difficult experimentally to take all this information in at the same time. So we need to be a bit creative statistically on how to fill in the missing gaps.
The history of this field has been that it has been very well supported. So that's led us to an extremely rapid rate of growth in how technology has developed. And that in turn has led to enormous changes in the sort of things we do. Nominally, when I came here, I could have retired. But I thought, this is such a great idea to try and do this institute. There's always interesting things to think about.